Hello, everyone. Welcome to part 4 of chapter 2. So, this one, we're going to be focusing on organic chemistry. We want to remind you kind of organic compounds. These are things that have carbon and hydrogen, contain carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. Chomps. They are covalently bonded together, so they will have covalent bonds between these different atoms to form these large organic compounds. Sometimes these are called macromolecules. Sometimes we call them that. They will have functional groups that will determine chemistry, give some of the function to them. They have a function. They participate in the chemistry and do all that cool stuff. Carbohydrates, that's one example. This be your sugars, things like that. Lipids, your fats, waxes, and oils. Your proteins, and your nucleic acids, that's your DNA and RNA. Now, there is a sub of these called high energy compounds, ATP, ADP. These high energy compounds are also found associated to nucleic acids. They are derived from nucleic acids, derived from RNAs. So. Sorry, there should be a title slide on this, but there's not. Where I want to talk about monomers and polymers. Monomers are the basic building blocks that make up polymers. Polymers, kind of think of a polymer, the monomer and polymer. Think about a train. Each train car is the monomer, and the entire train is the polymer. Okay? The repeating monomers join together through dehydration synthesis, and they form polymers. I'm sorry, there should be a title slide there. I'll have to edit that and adjust it. It's just something I ever did do before I posted the PowerPoints. I went through a couple little changes here and there over the winter break that I wanted to do some things to my notes. Anyway, so functional groups. There are lots of functional groups. What these things they are going to do is they're going to have functions. The ones I really want to worry about is the amino group, NH2, the carboxyl group, a COOH, a hydroxyl, an OH, and a phosphate, a PO4 2 minus. Now I put the 2 minus charge right here. They have the 2 minus, and you can see it here. So I have PO4 2 minus, they have PO4, but I put the 2 minus charge on it here. Okay. Let's talk about carbohydrates. They are really about less than 1% of your total body weight. They're an important energy source. We catabolize them. They are energy. Carbohydrates are the primary fuel for cells. One of the things about them is they're going to have a carbon-hydrogen-oxygen ratio of 1:1. That means for every 1 carbon, there are 2 hydrogens and 1 oxygen. Okay? Carbohydrates can include monosaccharides, my simple sugars, my disaccharides, which are 2 sugars, and my polysaccharides, which are many sugars, which is 3 or more sugars bonded together. Monosaccharides, which we call simple sugars, with they can contain anywhere between 3 to 7 carbon atoms. Examples of these, which I will expect you to know which of these are monosaccharides, disaccharides, and polysaccharides for a test. I may come out and ask you which of these is a monosaccharide, which of these is a disaccharide, which of these is a polysaccharide. Glucose, fructose, galactose, these are all monosaccharides. And then basically you can take, for example, glucose and a fructose and put it together and make a disaccharide called sucrose. Sucrose, table sugar, is glucose and fructose bonded together. 2 glucoses, that's maltose. A glucose and a galactose makes a lactose sugar. That's milk sugar. Polysaccharide, glycogen, starch, and cellulose, fiber. Glycogen, starch, and cellulose is just a bunch of glucoses. Okay? They form long chains. Now, something that I do want to describe, there are multiple ways to describe sugars, especially monosaccharides. One of the things that we can do is describe them by how many carbons they have. There is another way that we describe them. If you ever want to learn that, take me for general biology, and I'll explain that method as well, that biochemistry. But I don't worry about it for anatomy and physiology. That's a little bit, our book doesn't go into those. But our book does talk about these. Now, we have triose, tetrose, pentose, hexose, and heptose. Tri means 3. That means it has 3 carbons. Tetra means 4. It means it has 4 carbons. Penta means 5. It has 5 carbons. Hexa means 6. It has 6 carbons. And hepta means 7. Has 7 carbons. So we have between 3 to 7 carbons: triose, tetros, pentos, hexos, heptos. 3, 4, 5, 6, and 7 carbons. We can put them together to make a disaccharide, or put more of them together and make a polysaccharide. Okay? We can covalently bond them together. Now, the most important monosaccharide out there is going to be your glucose. Glucose is somewhere about is the most important one. It's your blood sugar, for example. When we say blood sugar, we are talking about your glucose. It has a chemical formula of C6H12O6. It can be shown as a linear chain or a ring structure. Ring is the most common form when in water. So that's why it's the most common form of glucose is a ring. Now, we can take these and put them into chains and form disaccharides or polysaccharides. Complex sugars, 2 or more. Table sugar is a disaccharide. Starch is a polysaccharide. Glycogen is a polysaccharide. We call it animal starch sometimes because it's an important storage polysaccharide. For muscle cells, for example, they use that. I don't have cellulose anywhere else mentioned, but I did mention cellulose previously. How we can make more complex sugars is, for example, here we make sucrose by taking a glucose and covalently bonding it, dehydration synthesis between the two, forming a glucose-fructose covalent bond, forming a disaccharide called sucrose or table sugar. Okay. Now, lipids, they are hydrophobic. That means they are water-fearing. They are things like fats, oils, and waxes, or fats, waxes, and oils. They are mostly made up of carbon and hydrogen atoms, so a lot of these are hydrocarbons, but not all of them. For example, your fatty acids are going to be hydrocarbons. Your cosinoids are going to be heavily hydrocarbons. Your glycerides are heavily hydrocarbons. Even your phospholipids and glycolipids heavily are hydrocarbons. Steroids have other things going on, but carbon and hydrogen makes a large composition of it. So these things are mostly going to be carbon and hydrogen. Fatty acids, eicosanoids, glycerides, steroids, phospholipids, and glycolipids. Lipids are mostly an energy source, but they can be energy storing, such as fat. So energy source is a big thing for lipids, but they have some other functions that I want to talk about. Now, fatty acids. One of the things I will talk about with fatty acids coming up here is some things about fatty acids that are very important that I'll want to talk about. Fatty acids mostly contain carbon and hydrogen to a 1:2 ratio. For every 1 carbon, there mostly will be 2 hydrogens. Mostly. Okay, not all of it. If you look here, this carbon does not do that. It's got 1 carbon and 3 hydrogens. And this carbon has got an OH and a carbonyl on it. It's got a carbonyl, so basically a carboxyl. So basically we have a fatty acid with a carboxyl group on one end. This is a fatty acid. Fatty acid is nothing more than a hydrocarbon, which is purely carbon-hydrogen chains with a carboxyl group, COOH, at one end. So what we have here, guys, is this one, CH3, but the rest of these mostly are CH2s. Okay, this one, for example, is called lauric acid. Okay, so they are nonpolar except for the carboxyl. The carboxyl group is polar. The rest of this is nonpolar. Carbon and hydrogen have a very close electronegativity, so they form nonpolar covalent bonds. Okay. So here's something I do want to talk about and help you guys understand is this idea between saturated and unsaturated fats. So let's explain this. Let's talk about the idea between, so if I take a carbon, for example, here, and I take these carbon chains, okay, like that. And every dash represents a single covalent bond, and the double dash represents a double covalent bond. What we will see is there is in this fatty acid chain here, there are no double bonds, okay? So what we will notice Let's do this. 1, 2, 3, 4. Let's put a double bond right there. Okay. All right. Now, what has happened is here and here we can't have hydrogens. So what we have made right here is a fatty acid, but right here there is a double bond. Okay. All right. This is called a saturated fat. This is an unsaturated fat. Sorry if my dog's snout ends up being in camera. He's licking me, trying to get my attention. Okay, buddy. Okay, sorry. It's the thing about doing this at home is my dogs are always going to be here. So, okay, so this is an unsaturated fat. Now, actually, these unsaturated fat, this is what is called a cis fat. If we had the hydrogen here and a hydrogen here, so if we did this, we would call that a trans fat. And if we have this, we call it a cis fat. Okay. You probably heard of trans fats. Okay. So that's actually the difference between a trans fat. That's what a trans fat is. Okay. All right, now let's talk about eicosanoids. Eicosanoids are a group of lipids that act as hormones, other cell signaling molecules. They are leukotrienes and prostaglandins. Leukotrienes are active in the immune system. They are made by leukocytes, hence leukotriene. Okay, leuko, they're from a leukocyte. They have 3 double bonds, so that's why we call them triene. Here we have 1, 2, 3 double bonds in the fatty acid tail. This means that what we have is a triene. When you have fatty acids, when you have hydrocarbons that only have single bonds, we call that as something ane, like propane, butane, heptane, nonane, octane, etc. Ane. If we have double bonds, we call it something ene, propene, octene, ethylene, etc. There's ethane, ethylene, ethylene. So, for example, 1, 2, 3 double bonds. 1, 2, 3 in this ene. Ine is triple bonds. If you have single bonds, we call it ane. If this has double bonds, ene. If it has triple bonds, ine. That's just some basic organic chemistry. This is why we call them that. You don't have to know that. It's just I'm teaching you that because I want to explain to you why it's called triene. It has 3 double bonds in it. A prostaglandin being a local hormone means they work where they're released. They are short fatty acid chains. There are 2 of them, and there's a 5-membered ring. That 5-membered ring and 2 fatty acid chains make them a prostaglandin. They're involved in pain and inflammation. Prostaglandins, pain and inflammation. Pain, prostaglandins, okay? Now, glycerides, these are attached to a glycerol. So we have a glycerol molecule with fatty acids. Triglycerides have 3 such fatty acids. Diglycerides have 2, monoglycerides have 1. They are important energy sources. They act as insulation and protection. We call them triglycerols or neutral fats as well. Steroids, they are going to have 4 fused rings. So notice 1, 2, 3, 4. They all have this basic skeleton of 4 fused rings. 3 of these are 6 carbon rings, and 1 of them is a 5 carbon ring. So they all have fused rings, 3 6 carbon rings, 1 5 carbon ring. 3 6 1 5, 3 6 1 5, 3 6 1 5. Okay, 3 6 carbon rings, 1 5 carbon ring. They all have it. Cholesterol, estrogen, testosterone, for example. Cholesterol is an important thing found in your cell membranes and found in blood, a component of plasma, cell membranes. It's found in the blood. Plasma cell membranes, okay? Plasma membranes or the plasma membrane or cell membrane. Estrogen and progesterone, testosterone, the sex hormones, they make that. They make your corticosteroids and your calcitriol. They're metabolic hormones, metabolic regulators, and bile salts even are derived from steroids. Basically, if you want to lower cholesterol, what you do is eat a diet high in fiber. The fiber binds to the bile salts, and then you defecate the bile salts instead of reabsorbing them, and your body will take cholesterol from the blood to make new bile salts, and that's why eating a high fiber diet lowers your cholesterol. All right, phospholipids and glycolipids. We call these diglycerides because they only have two fatty acid chains. They have 1, 2. Now, as I was talking about that double bond, cis versus trans fats, right here, when you actually have what is called a cis fat versus a trans fat, cis fats are like this, where they have a little kink in them. Trans fats don't, they're more linear. So cis fats tend to be more liquid at room temp, okay? Oops, sorry. Okay. Now, they have hydrophilic heads that are charged, and they have hydrophobic tails. The head has charges to it, whether it's a carbohydrate or whether it is some non-lipid groups like phosphates or this choline right here. This is choline, this is a phosphate. Okay, so that's what's going on there. Okay, they can so they have hydrophilic heads and hydrophobic tails. They're important they're important components of your cell membrane. All right, now proteins. Proteins are very important. They are the jack of all trades. They do so many things. There is nothing that does more jobs than proteins. They can be support, structural proteins like collagen. They can be movement. They can be contractile proteins like the muscle proteins. They can be transport proteins or carrier proteins like hemoglobin. They can be buffering proteins. They can regulate the pH. Proteins can act as buffers. We'll learn about that in AP2. We'll learn how hemoglobin acts as a buffer. We'll learn how amino acids act as buffers, amino acids, proteins. We metabolic regulation, for example, enzymes, coordination and control, your hormones and neurotransmitters, many of them are proteins, and defensive proteins like antibodies are defensive proteins. And there's various other kinds of proteins used in defense, like that we'll talk about in AP2. I wish we got to talk about some of the other proteins that act as defense, some really fascinating things that I enjoyed to talk about, but our book doesn't go into them. Okay, so let's talk about what makes up your good little amino acids. Amino acids are the building blocks for proteins, okay? So proteins are made up of amino acids, okay? So amino acids. So amino acids are going to be what makes up proteins. To make an amino acid, one of the things we're going to need is a carbon found at the center here. Carbon has the capacity to form 4 covalent bonds. Okay, we refer to that as being tetravalent. Okay, now because carbon is tetravalent, it likes to bond to some things. One of the things it likes to bond to is an NH2. Now, it likes to bond to that amino, which is called an amino group. It likes to bond to a COOH called a carboxyl group. It also likes to form with a hydrogen. And what this creates is a backbone. Okay? Now, what it also likes to bond to is a group that we are going to make a stand-in for R. R is going to be a stand-in. It can be anything we want it to be that can bind to it. If it can bind to a carbon, it will bind to the carbon if it can bind to it. Any chemical, any group, it can be just a hydrogen. It could be some other kind of, it could be really simple, or it could be other things. It could be benzene rings, et cetera, et cetera. It's really complex things or really simplistic things and things in between. We call that an R group. It's this R R group right here is going to make every single amino acid differ. In the backbone here, every amino acid is the same. But right here at the R is where every amino acid that we ever have differs. Now, there's only 20 amino acids that life uses. Life uses 20 of these amino acids. So all amino acids have a central carbon. We call that the alpha carbon. They have a hydrogen atom. They have an amino group, and they have a carboxyl group, sometimes called a carboxylic acid group or a carboxyl group. And I'm just going to say carboxyl group. I'm going to change it to my notes to say carboxyl group. Now, there is an R group, and that's where they all differ. That is the place all amino acids differ. Now, if we look at this right here, here we actually have 20 amino acids. The side chains can be here yellow, green, the R groups, purple or blue, or pink and blue. Okay, so they are these different colored things. Those are all different R groups. Every R group is different, okay? Sorry, I keep hitting that. That way. Okay. Now, amino acids, they get bonded together. Now, when they get bonded together, they form what is called a peptide bond. That is the special name for the covalent bond that happens between the carboxyl group and the amino group. Forms a covalent bond through dehydration synthesis, forming a dipeptide. This dipeptide, if it's two amino acids, we form a dipeptide. If it's more, we call that a polypeptide. Okay. Now, this can be a little tricky to learn at first. This can be a little tricky, and I want to help you guys understand it. So this can be a little tricky and harder to learn at first. So let's help you learn it, help you understand it. Now, to get this down, we have to know that proteins can have 4 levels of structure. There's only one that all of them have. Some of them may have all of them. Some may have 2 of them. Some may have 3 of them. They may have all 4. They may only have 3. They may only have 2, but they have to have one. What do they all have to have is primary. Primary is the amino acid sequence. You can, for a polypeptide, that is the amino acid sequence. You cannot be a polypeptide, a protein, and not be made up of amino acids. Polypeptide and protein aren't really what we would say synonymous. A polypeptide is basically a protein is a polypeptide with a job, with a 3-dimensional structure that gives it a function. A structure that gives it a function. Structure and function are heavily related to each other. Okay. Now, your primary structure, that's your sequence of amino acids. Your secondary structure, this is when hydrogen bonds without R groups. This is usually fibrous in structure. It can be an alpha helix. That's if you take every 4th amino acid residue or constituent, every 4th amino acid, if you bond hydrogen bonds every 4 amino acids, then you will have an alpha helix. If 2 or more polypeptides are lying side by side and they connect through hydrogen bonds, you form a beta pleated sheet. A tertiary structure, that is when you have your R groups interacting, and they are normally globular. We mean by that, we mean more spherical in shape. Quaternary is when I have 2 or more polypeptides and I put them together. Okay. So we can have proteins that have all 4, and I'll show you. So here we have the primary structure, each amino acid representing part of the primary structure. Then we come every 4th amino acid and we bond it together and we form an alpha helix. Beta sheet comes out when we go side by side. Here we have our amino acid coming and folding in on itself, and side by side parts are bonding. Then, so here we have an alpha helix found in this tertiary structure. So we have an alpha helix in this hemoglobin tertiary structure. Then we take this tertiary globular subunit, stick 4 more together that are much similar to each other, and we have 2 that are identical, and then 2 other. We have 2 alphas, 2 betas. We put them together, and what we form is a hemoglobin. Now, this hemoglobin here, see how it has, hemoglobin has, if I look at this hemoglobin protein, it has 1, 2, 3, 4 individual polypeptides. This makes it quaternary. I look at it and I see it's made up of 3 tertiary subunits, 3 globular pieces, so it has tertiary structure. I look at it and see an alpha helix, so it has secondary structure, and it has to have primary because it's a polypeptide. If I look at this collagen protein and I look at it, it has to have primary because it is a protein. It's made of amino acids. I see secondary because I see an alpha helix. I don't see tertiary because it's fibrous and not globular. And it has quaternary because I see 1, 2, 3 polypeptides. Okay? So this hemoglobin that carries oxygen in the blood and this collagen, which is a fibrous connective tissue protein, this is what makes your connective tissues, is a lot of collagen. Okay? So here's what happens. Proteins, the primary structure will fold up and become secondary. You'll get your helices and your sheets. Then it'll fold up even more, and you might get a tertiary. You may get it, so you have to have primary. You may have a secondary if the chemical composition allows for it. You may end up with a tertiary. For example, this protein here has got primary, secondary, and now it's going to go into tertiary, and that's as far as it goes. It doesn't go any further. Okay? Here we have a protein with 4 different polypeptides, the yellow one, the blue one, the green one, and the pink one. These are 4 different polypeptides. We can see that it is a protein, so it's made of polypeptide, so it's polypeptide, so it's made of amino acids, so it has primary structure. I see alpha helices, so it has secondary structure. There's globular structure to it, so it has tertiary. And it's got 4 different proteins, so it's 2 or more polypeptides, 4 or more polypeptides making this one protein. So this protein here has 4 polypeptides to it, so this is quaternary as well. So it has all 4 structures. Okay. So globular proteins are rounded and they're water soluble. They're more compact and they're based on their shape. They are usually going to be enzymes, hormones, and proteins that circulate through blood. Globular proteins allow them to stay in solution because the hydrophobic parts are inside and the hydrophilic parts are outside. The shape of these come from their tertiary structure. Fibrous proteins are strands or sheets. They're usually tough and durable, but they're not water soluble. They are primarily from the secondary structure. Their primary secondary or even quaternary structures can give some of the fibrous structure, like collagen has multiple secondary subunits giving a quaternary structure that is highly fibrous as well. So like multiple strands of rope. So they are structural proteins. Okay. Now, the structure of shape of a protein determines its function. Very important. All proteins have a job, and to do their job, they have to have the right shape to do that job. The 20 different amino acids come together in many different ways to create an amino acid sequence, a primary structure that can maybe create a secondary structure, even a tertiary structure, and maybe a quaternary structure, depending on what kind of protein you are, if you have all 4 or don't, if you have primary, secondary, tertiary, if you have primary, secondary, quaternary, whatever, you're going to have at least primary. You're going to have a shape. That 3-dimensional shape, is going to give you your function. If you change by one amino acid, this can have huge impacts. One of the examples I like to give is I change one amino acid, just one amino acid, in hemoglobin's beta subunit, and this causes dramatic change to beta hemoglobin structure. This changes the quaternary structure of hemoglobin. This changes the red blood cell structure in people who have sickle cell anemia. In sickle cell anemia, it's a 1 amino acid change that creates a dramatic change to hemoglobin structure that changes the cell structure that causes anemia, sickle cell anemia. Now, you can change protein shape by changing the salinity, the temperature, or the pH. If ions concentration gets off, these ions tend to go in and mess with the intermolecular forces that hold together the secondary and tertiary structures. They are temperature, it's going to mess up the bonds. The hydrogen bonds that form are going to break. pH, it changes up the intermolecular forces that hold it together. The chemical bonds that can hold proteins together can be disrupted by ions, temperature, or pH. Get it too salty, too hot, or too acidic or basic, and it messes up the protein structure. Okay. Now, proteins can oftentimes be enzymes. Enzymes, remember, are catalysts, and what they do is they lower the energy that makes a reaction go, the activation energy. I describe this like getting a little coupon to or a gift card to your favorite restaurant. So if you really like to go, but it's kind of pricey, and you don't want to have to pay that much to go to this place, but you really like it, and someone gives you a gift card that pays for half the meal, then you're likely to go because they've reduced the cost. If we reduce the energy that is needed needed to make a reaction go, we've basically saved that energy. So enzymes are proteins that lower activation energy. They usually are going to be proteins. They're not always, but they usually are. For example, a ribozyme, it's going to have an RNA-based enzyme. But they are also not changed or used up by the reaction. So an enzyme, when it works, it can do it again and again and again and again. Okay, they're reusable. They're able to continue to function for quite a long time before they get peed out or removed somehow by the body. But enzymes are very specific. They only work on certain substrates. Enzymes are very limited by how much there is. There is only so many active sites that can be binding to substrates. So they're limited by how many active sites they have. They're also highly regulated. We take chemicals like ions and vitamins and we regulate them. Okay. The active site is where the substrate binds. That is the reaction site. That is the binding site for substrates. The substrate is the substances, the reactants that react with enzymes. The enzyme substrate complex is when we take the enzyme and its active site and we bind them together. And we have the active site of the enzyme with substrates bound to it. That is called the enzyme substrate complex. And then whatever is produced by the end of the enzyme reaction is called the products, okay, or the product. So we have substrates come together to bind to an enzyme's active site. The enzyme is like a molecular matchmaker in some cases. It can be like a molecular matchmaker. It can put substrate 1 and substrate 2 together in a favorable way and make a product that is bigger. These can be, for example, these are enzymes that build things up. This would be an enzyme that is what we refer to as a anabolic enzyme. This enzyme is doing anabolism. It's building things up. It's synthetic. It's doing synthesis. Now, we mentioned that they are aided by things. There's cofactors and coenzymes. Cofactors are molecules that can bind to an enzyme to change it. It binds to it like an ion or other molecule, a cofactor. A coenzyme is basically an organic cofactor like a vitamin. Our vitamins are necessary for certain enzymes to function. There's enzymes, for example, an enzyme that is needed to make collagen, part of the biosynthesis of collagen, there's an enzyme that needs vitamin C to function. If you don't have vitamin C, you don't make collagen. You don't make collagen, you get a disease called scurvy. So that's caused by a vitamin C deficiency. You don't get collagen being made, your skin gets weak, your teeth fall out, scars open up, and all kinds of things like that. Sores, issues like that. Skin tears open, teeth fall out, stuff like that. Proteins can lose their shape due to temperature, pH, and salinity. That is called denaturation. It changes the shape, you change the function. Remember, form fits function. The structure of a protein is determined by its shape, okay? All right. Now, lastly, we're going to talk about nucleic acids. Nucleic acids, they're found in the nucleus of a lot of your cells. They're a very large organic molecule. They're found heavily in the nucleuses of cells. They store information molecularly. This is genetic information. They act as a molecular storage device. This involves DNA, deoxyribonucleic acid, which is what your inherited characteristics are coded by. They direct protein synthesis. They control enzymes. They control metabolism. So genetics can grace you with favorable metabolism to keep yourself skinny and slim and things like that. But now I was always very skinny as a kid, and then I hit 40, now I have to watch my weight. Now, ribonucleic acid RNA, that is the intermediate go-between. There's, for example, messenger RNA, transfer RNA, and ribosomal RNA, mRNA, tRNA, and rRNA. These are special types of RNA. Now, RNA, DNA, nucleic acids, they are made up of strings of little building blocks called nucleotides. The nucleotides are the smaller building blocks that make up DNA and RNA. All nucleotides have something in common. They all have a 5-carbon sugar called a pentose. If you are DNA, this sugar is deoxyribose. If you are RNA, this sugar is ribose. It's either deoxyribose for DNA, ribose for RNA. They all have a phosphate PO42 minus, and they all have nitrogenous bases bound to them. The nitrogenous bases can be G, T, C, A, or U. Okay. Now I always just say GTCA. I know it here says AGTCU. I say GTCA or U. It doesn't really matter that I'm reading it in order. It's GTCA is just a kind of an easy way for me to remember it. So, okay. Now, these nitrogenous bases come in two types, purines and pyrimidines. Purines have two rings. One of them is 5-membered, one is 6-membered. But I want you to remember this more than anything. Is if you're pure, you're all good. Adenine and guanine, all good, are purines. Adenine and guanine are purines. Now, all good. If you're purine, you're all good. Pyrimidines, what do you do with pi? You cut the pi. Cytosine, uracil, thymine, CUT, cytosine, uracil, thymine. DNA, thymine, cytosine has Y. RNA, uracil is unique only to RNA. Now, cytosine has a Y, but uracil doesn't have a Y. Uracil is only RNA. So DNA has thymine, RNA has uracil. Both DNA and RNA have cytosine. But thymine is unique to DNA, uracil is unique to RNA. Both have cytosine. Pyrimidines you cut pi. Okay? So you can see purines, adenine, guanine, pyrimidines, cytosine, uracil, thymine, cut. Thymine is DNA only, uracil is RNA only. You definitely need to know that. Okay, it's going to be on a test. Something about that. Okay, DNA is what we call double-stranded. We call it a double helix. It is double-stranded molecule that is bonded together with hydrogen bonds. One strand here, okay, has so many bonds, okay? The A's and the T's, T and A, have 2 hydrogen bonds between it. C and G have 3. CG3, TA2, okay? CG3, TA2, okay? Now, they form what we call base pairs that are what we say complementary, where we always see adenine and thymine always bonding together. T and A go together, A and T. Apples on the tree. Cytosine and guanine go together, car in the garage. Apples on the tree, car in the garage. Apples on the tree, adenine and thymine bond together. Car in the garage, cytosine and guanine go together. Adenine, thymine, TNA2, cytosine, guanine, CG3. Okay. RNA has uracil, which replaces thymine. So uracil of RNA would pair with an adenine if when RNA pairs with DNA. Okay. All right. Last thing we're going to talk about is ATP, and then we're done. Okay. ATP is a high energy compound. We're going to talk about adenosine triphosphate. Okay. But we want to start off with adenosine monophosphate. It has one phosphate, no high energy bonds on it. If we add a phosphate to that, we get 2 phosphates called adenosine diphosphate. Adenosine diphosphate has 2 phosphate, di meaning 2, and now with ADP, AMP, ADP, diphosphate, 2 phosphates, we get 1 high energy bond. With adenosine triphosphate, we have adenosine, which is an adenine, and an adenine and a ribose. An adenine and a ribose make adenosine. Adenosine plus 3 phosphates is ATP. Adenosine plus 2 phosphates is ADP, and adenosine plus 1 phosphate, AMP. Okay. Now, a good way that I kind of give my students to remember this is with ATP. ATP, there's 3 phosphates, and there is 2 high energy bonds. ADP, there is 2 phosphates and only 1 high energy bond. And AMP, there's 1 phosphate, no high energy bonds. Okay. Now, once we have those high energy bonds and we break those high energy bonds and release a phosphate, energy is released. This is because ATP is our primary energy source. When the high energy bonds are broken, the energy releases. This energy is making ATP potential energy. When we add a high energy phosphate group to a molecule, we break the high energy phosphate off, we give it to a molecule, make it do things. The fancy word for this is called phosphorylation. Okay. In micro, you'll talk about substrate level phosphorylation or an oxidative phosphorylation. Okay. Guys, this finishes our chapter. So thank you for being patient and going through 4 lectures on this. Thank you, guys. Your test covers chapters 1 through 2. So this is for your test next week. Okay. Thank you, guys. And if you come to study group, I will give you extra help. Thank you and have a great day.